So now I'd like to call Cassandra up to the podium to initiate our historical panel. And let me tell you something.
Okay. Thank you very much. And so I made a deal, exactly, whereby I could advance my own slides. So we're going to stick to that deal, right, guys? Thanks. Okay. Thanks, all of you, for sticking with us this long. Hopefully, I know that we are the last thing standing between you and your drinks. So we'll try to keep this entertaining. My name is Cassandra Extavour. I work at Harvard University, and I have never worked on Hox genes myself directly. However, I have observed them indirectly, up close with some of the people who worked on them, and it's my great pleasure and honor to be able to allow these three pioneers in this area to tell you a little bit of their story firsthand, and then to ask them some questions that I personally think are very interesting, and that I hope you will also think are interesting. So before we bring them up, let me just bring everybody up to speed. I don't need to explain what a Hox gene is, but I think it's interesting for us to remember where these things come from. And in case you don't, can you see my mouse? I have no pointer. That complicated drawing is supposed to be a homeodomain interacting with DNA, which we know is what we're supposed to do. All right, so I'm left-clicking. There we go. So William Bateson, of course, we are supposed to owe a debt of gratitude to for coining the term that gives its name to this meeting, for coining the term genetics to describe the study of heredity and of hereditary variation. And he was very interested in hereditary variation, in other words, evolution. And he documented variation in phenotypes that he observed in the animal kingdom in this book published in 1894. And in this book, he coined another important term. Genetics was coined a little bit later, but here he coined the term homeosis, which is where we now take homeotic genes, homeotic transformations, et cetera, from. And homeosis he defined as morphological changes whereby something has been changed into the likeness of something else. Some part of the organism is mimicking some other part. Okay, so this is where the name came from for these genes that we study. Is it 3 right clicks per slide, or what am I supposed to do here? All right. So here's a picture of the man himself, William Bateson, and 2 important scientists in the 70s took direct inspiration from his work. The first of these was the late Nobel Prize winner Ed Lewis, who will appear on the 3rd left click of my right index finger, or you could please advance the next slide for me. So Ed Lewis, working at Caltech for many years, making many seminal contributions not only to developmental genetics but also to important concepts in evolutionary biology, studied a complex of genes, now known as the bithorax complex, containing many individual loci. Next slide, please. That each had a number of separable phenotypes, and he was able, using these studies, to suggest that segmental identity of different regions of the body could be controlled in potentially stepwise in an anterior to posterior manner by genes within this gene complex. And that was an important insight to have, not only with respect to the global regulation of body patterning, but it also had evolutionary ideas in it that, of course, the realization of those ideas in the revelation of the conservation of homeodomain of homeobox genes and their conserved role in regulating animal body plans are among the things that earned him the Nobel Prize years later. Antonio Garcia Bellido, my PhD advisor, visited Caltech on a number of occasions, first in the 60s and then in the 70s, and through those visits to Ed Lewis's lab at Caltech became introduced to the bithorax complex. And Antonio was always very interested in the study of development at the level of cells. He wanted to understand how individual cells did things. And so what he did with genes in the bithorax complex was to do things like cellular dissociations and mixing of cells that were bithorax loss of function with wild-type cells and see how they behaved, or make clones of loss-of-function mutations in the bithorax complex loci and observe how these individual cells behaved. And by doing this, he observed that cells that suffered from these mutations behaved developmentally just like the normal developmental cells of the type of organ that they were mimicking. And so because the developmental trajectory of the homeotic transformation at the cellular level appeared to exactly resemble the wild-type developmental trajectory of the mimicked body region, Antonio thought that perhaps what this meant is that body identity, one, was controlled in a cell-autonomous way depending on the genetic makeup of the cell, not of the physical location in the body that it came from, which might speak against models that rely entirely on extrinsic information given to naive cells, such as, for example, morphogen gradients, if these things actually acted in a vacuum. Okay, so that's one important way that Antonio was thinking about Hox genes. And the other thing that he said, perhaps we can deduce from these cell autonomous phenotypes that there are certain types of homeotic genes that act as what he called master selector genes that then control region-specific developmental genetic cascades that function in a stepwise fashion. All right, so we had at this stage then, at the end of the 70s, a lot of phenomenology about what cells and organs and body regions did when they suffered from these different mutations that we're calling homeotic. But we had no idea really how things were working at a molecular level. And of course, to even say molecular level, molecular biology was just being born itself at that time and not necessarily always being taken seriously by all scientists. So here we have to acknowledge, before we get to our star panelists, the people who laid the molecular groundwork, I would say, within which they were able to conduct their studies and make their important breakthroughs. And so I want to acknowledge here these three key mentors in whose laboratories the raw material in the molecular sense was able to be provided. In the lab of Tom Kaufman at Indiana University, we saw in 1983 the publication of the molecular characterization of the Antennapedia complex. In a parallel paper in the same year from the Hogness lab, we saw the molecular characterization of the BXC complex. And using these sequence data in the lab of both Tom Kaufman at Indiana and in Walter Gehring's lab in Basel, our three panelists were able to use these sequence data and observe something very fundamental, which is that genes from both of these complexes, which by classical genetics we knew were physically far separated on different chromosomes at least, genes from these different complexes that had analogous developmental phenotypes, they were homeotic genes, also had homologous sequences in them. So the three people who you're going to be hearing from are Michael Levine, who's currently working at Princeton. Michael did his undergraduate work at Berkeley and his PhD at Yale, and he's currently the director of the Louis Siegler Institute at Princeton. Bill McGinnis, who did his undergraduate studies at Berkeley and San Jose, and is currently the Dean of Biological Sciences at UC San Diego. And Matt Scott, who, Matt, I've forgotten where you did your undergrad. MIT, I've heard of that place. They have a round building. And has fairly recently become the director of the Carnegie Institute. And so these people, using those sequence data that were generated in these labs, in sort of 1 year to the next, we have molecular characterization of these things. We have detection for the first time in whole mount in situ hybridization in 1983 of the spatiotemporal patterns in embryos as they are being patterned of these genes. And then we have the recognition by these scientists that these genes share not only analogous developmental functions, but DNA sequences that translate into homologous protein sequences that are going to have homologous molecular functions. And furthermore, that these special DNA binding sequences, as we later realized they were confined not only to homeotic genes, but also to other genes in Drosophila that were not homeotic genes, like Fushitarasu, for example, and also to many genes in other animals. All right. So with that introduction, let me turn it over first to Mike Levine, who's going to tell you a little bit how he ended up being at Basel and what were the steps that led him to do the work that he did. All right. Absolutely.
Thank you very much.
So just be persistent with the clicking, or
Okay,
just do that.
Thanks a lot, Cassandra. I got to tell you, it's very weird, okay, being asked to give one of the old fogey talks. I'd rather give a real seminar, but apparently I have no new data, so we have to go back in time approximately 33 years. And let me just say at the outset, I don't know what these other two bums are doing here. I had all the ideas, okay? I just want you to remember. Okay. So if I can have the next slide. Can I have the next slide? All right, so I arrived in Basel, Switzerland in August of 1982 to begin my postdoc with Walter Gehring in the middle there. And I immediately forged a collaboration with a graduate student, Ernst Hoffen, who had been working on in situ hybridization methods to detect, for some reason, heat shock transcripts. And so I said, look, Ernst, let's do something interesting. Let's further develop the method on tissue sections and look at some interesting genes. And fortunately, just a few months after we started our studies, another postdoc in the lab, Rick Garber, cloned the homeotic gene Antennapedia. And this was done independently of the work that you'll hear about from Matt Scott later on. And by the late fall of '82, we were beginning to see the first localization of Antennapedia transcripts in sectioned embryos on the bottom there. That's a mid-staged embryo. and the arrows on the bottom point to antennapedia transcripts in the mesothoracic, the middle thoracic ganglion, T2. And that was very exciting to us because we knew from genetic studies that antennapedia gene activity was primarily restricted to the middle thoracic segment. And that's based on studies from Tom Kaufman's lab, Gary Struhl, and others. Now, at around the same time that Ernst and I were doing the localization studies on antennapedia, if I can have the next slide, Michael Acom, who was starting his own lab at Cambridge University, was looking at the expression profile of UBX. Now, Michael had been a postdoc in the Hogness lab, and he obtained some UBX probes from Welcome Bender and Pierre Spear, who did that Herculean chromosome walk through the UBX locus. And Ernst and I had Acom out for an informal seminar in the spring of '83, and he showed us some of his early localization studies with his UBX cDNA probe, and we were all very struck by the similarities in the antennapedia and UBX expression patterns. Basically, they show the same tissue profiles, but the main difference is that antennapedia was shifted one segment in front of UBX. So antennapedia transcripts were detected in the middle thoracic T2 segment, whereas the UBX transcripts were detected in the T3, the metathorax, and the first abdominal segment. But other than that shift, spatial shift, the two patterns were remarkably similar. So this made us think, you know, maybe somehow the genes might be related. And this idea that they could have some relatedness was reinforced by evidence that the genes seem to regulate each other as well. So if I can have the next slide, and I only have this and one more. This shows the expression of antennapedia in a mutant that lacks all the genes of the bithorax complex. And I got to tell you, I had a secret weapon during my short time in Basel, and his name was Eric Frey. He was my roommate and one of the most brilliant people I have ever encountered, an incredibly creative guy. And we talked a lot, you know, we were roommates. And he made the critical suggestion that Hoffen and I should put the antennapedia probe on different bithorax mutants. And that's what you see in the lower right there, is the distribution of the antennapedia transcripts in a mutant embryo that's homozygous for the famous P9 deficiency, which lacks all the genes of the bithorax complex. And you can see that instead of the antennapedia transcripts being restricted to the mesothorax, you see very strong expression throughout the more posterior regions. So this was very early evidence for the so-called posterior prevalence rule that was popularized by Denny DeBoule in his studies in mice years later. But the main thing is that this is evidence that the posterior genes, acting genes of the bithorax complex, somehow repressed the anterior homeotic genes such as antennapedia. So we have antennapedia UBX showing very similar patterns of expression and evidence that they regulate one another. So we really felt like the genes could be related. And of course, this idea didn't come right out of a vacuum. I mean, it was out there in the scientific culture. If I can have the next slide. And it was, one back. One back, there it goes, there goes the thunder. Okay, in his famous '78 paper, oh, come on. There you go, that's it, that's all right. I know you guys are expecting a bevy of like Jewish jokes, but look, I'm a director of a fancy institute at Princeton, and I'm not allowed to be, I can't do that anymore. Okay, I got my little preppy sweater on, and okay, anyway. So in his '78 paper, Lewis, I mean, you know, that paper, I don't know about you guys, that paper was really hard to read. It literally physically gave me a headache the 60 or 70 times that I read it. But early on, there's a very lucid sentence there. During evolution, a tandem array of redundant genes presumably diversified by mutation to produce the bithorax complex. Now, that's straight up. These genes are related to each other. And then Tom, in his 1980 paper, basically proposed that the antennapedia complex was an extension of the bithorax complex. It was patterning the anterior segments in the same way that the bithorax genes were patterning the posterior segments. And so, altogether, the in situ data, the papers from Lewis and Kaufman, 1978, 1980, really suggested that these genes might be related. And Ernst and I were very fortunate that in March of 1983, Bill McGinnis joined the search for relatedness among the homeotic genes. And if I can have my last slide, Bill took the enterprise to another level. Next slide, please. Okay. What is going on? All right, here we go. Now that's a picture, a contemporary picture from the time, and I'm showing Bill one of my shitty gels, and you can see by the look on his face, he's not too impressed. I think it's fair to say that without Billy, the Homeobox would not have been found in the Gehring lab. So I'm going to turn it over to Bill.
So I came to Walter Gehling's lab, and initially my plan was I wrote a fellowship proposal on trying to clone new homeotic genes by using transposable element tagging. But it became clear after about a week in Walter Gehling's lab that it wasn't the place to do it. And so I gave that up immediately. And there was a guy working in the lab, Bernard Meckler, who wanted to clone and study the lethal giant larvae gene. So I got together with Bernard, and I'm sorry, Bernie, if you're here, I'm using Bernard. By the way, I would like to say that every scientist that we've seen after Bateson looked happier than Bateson in that picture. And the idea that we had is we'd make some southern blots with some DNA from different species of Drosophila, virilis, Heidii, and so forth, because we didn't know which sequences in the lethal giant larvae gene were important, and we'd try to see if we could find some conserved sequences that would hybridize, and we'd focus in on those sequences. Well, that didn't work very well, and I was having lunch with Mike and Aaron Stoffen one day, and Mike said, well, why don't you use the Antennapedia cDNA and try this? At the time, Mike thought this was a much more interesting gene, although, as we saw earlier, lethal giant larvae is equally as interesting. And what? And within a very short period of time, so we hybridized this Antennapedia cDNA to some southern blots with different Drosophila DNAs. And the initial results were ugly. And that's one of the things I want to emphasize here is the role of kind of chance and contingency and how things look great in retrospect, but when you're prospectively doing it, things seem horrible and ambiguous and like you're going to fail. In this particular case, we didn't fail, but certainly it happens many times for us. So and the next blot shows the next slide. On the next slide. On the blot on the right-hand side shows one of the first southern blots that we got using some probes, small probes. So when I was a grad student in Berkeley, I found out how not to do low stringency hybridization because I was trying to clone different glue protein genes, which I could never have cloned because they weren't a gene family. But I did learn some lessons about how not to do low stringency hybridization. And so some of those lessons were useful in seeing some of these banding patterns where, for example, in the two right groups of bands there, you can see that there's about 8 genes or 8 DNA fragments or so that hybridize with a probe from the Antennapedia gene, which was a small probe that we figured out had a low frequency repeat because there was another part of the gene that had a high frequency repeat. And with those genes, we thought, well, you know, that looks roughly like the number of homeotic genes in the fly genome that we knew about. Actually, we thought it looked too low because at the time we thought that there was probably 10 or so homeotic genes in the bithorax complex. So it didn't look that good. But we thought we would isolate those different DNA fragments. And that proceeded very rapidly. And what Mike and Ernst and I did was isolate those fragments in in clones of lambda, large DNA fragments, hybridize them to polytene chromosomes. Many of those clones hybridized to the Antennipedian bithorax complex. For those clones, we just went right ahead and did in situ hybridizations. It was very simple, straightforward, took almost no time at all, a period of weeks, and it was a wonderful time where you just cranked the experimental apparatus and the results came at. It was fabulous. And eventually it resulted in this picture where after a couple years later, we could identify those different bands that were hybridizing with this Antennipedia probe as indeed two of the bands were hybridizing that were part of the Antennipedia homeotic gene. Fujitaratsu, the gene, the segmentation gene, but let me remind you, Fujitaratsu about 300 million years ago was a homeotic gene. It just changed recently. The deformed gene, abdominal B, ultrabithorax, sex-reduced abdominal A, all homeotic genes of the Antennipedia complex and bithorax complex. And so we published all this very rapidly. But I do want to say that during this process when we were isolating these clones with these probes, whenever you're isolating genomic fragments, genomic clones, you isolate a lot of different clones. And not all of them actually had strong homology to these fragments. And so you'd have a lot of clones. We'd do the in situ hybridizations to the chromosomes, and you'd see, well, this doesn't map to the Antennipedia complex or bithorax complex. Where does it map? And at one time we had 3 or 4 of these clones that we'd isolated that mapped to the cytological location of tRNA genes. And so Michael and I spent like a week, we're cloning tRNA genes. Who cares about this? Our careers are over. So there was a lot of ambiguity at the time. It looked great in retrospect, but it was sometimes seemed very ugly at the time. One thing that gave us a lot of confidence early on, and you also have to realize we didn't know what these coded for. We didn't even call these homeoboxes at the time because we didn't know that they encoded protein coding sequences. They were just fragments that hybridized with Antennipedia and Fujitaratsu. And it wasn't for some time that we knew that they actually encoded for homeodomains. At the same time, even though we didn't know what we were working with, we started to do some so-called zoo blots to see whether those sequences, if they were homeotic coding sequences, if they were conserved in other animal genomes. So we go to the next slide, please. And this was the team. I just wanted to show you also a contemporaneous picture of the team of Ernst Hoffen, Mike Levine, and me, who were working in a wonderful collaborative relationship, talking to each other every day, challenging each other every day, tormenting each other every day, listening to Mike's ridiculous theories every day and rejecting many of them, but listening carefully. And it was just tremendous fun, the time we had then. The funnest time I've ever had doing science. But the next thing that Mike and I did, so Mike was fabulous at making hot hybridization probes. In fact, his habit was to use as much radioactivity as he could in a reaction. He would use all the radioactivity in the lab. So much that the probes, that everything would leak all over the bench. So there'd be a radioactive bench. Now, Mike didn't want to clean up the bench, so he would move to another bench to do his next in situ hybridization. And it would be up to Ernst or me to clean up that bench so that Mike could do his next, he could keep transferring throughout the, that irritated some people in the lab, but. So the next slide shows signals, and I'm showing the raw data here, the old ugly data, because it gives you a feeling of what it's like to see the early results. So this was one of the first zoo blots we did with Homeobox probes. What we didn't know were Homeobox probes. They were probes that we thought were from Hox genes, homeotic genes, showing that there were some initial signals that calf DNA and frog DNA and some other vertebrate DNAs had sequences in their genomes that would hybridize with these Hox-derived sequences, these sequences that came from the coding regions. And one of the things that gave us confidence, so we were still worried all the time, and, you know, we weren't finding enough bithorax complex genes, why weren't we? And so luckily, I met Inez Morata at a meeting in Switzerland, and we said, look, you know, because we'd done some experiments with Francois Karsh and Welcome Bender, and we said, you know, we can only find 3 Hox sequences in the bithorax complex. And Inez says, well, that's perfect. There should only be 3 Hox sequences in the bithorax complex because we've only found 3 lethal complementation groups in the bithorax complex. And that also was a thing that even though we didn't even know what the sequence encoded at the time, it gave us confidence that we were on the right track. So all these little hints that helped us keep going. And then this led to Eddie de Robertis' lab was right across the hall, and so I gave a lab meeting to Eddie de Robertis, and then Andres Carrasco and I in Eddie's lab could clone the first Xenopus Hox gene, sequence it, and it turned out to be similar to the Drosophila Hox genes. And that led to, of course, the cloning of many other mammalian Homeobox genes. So I'm going to end there with the science, and then as far as stories, I hope to tell a couple later. Now I'll turn it over to Matt. Stay up here.
Yes, sir. All right. They're gonna drive? All right. Good. Well, hi, everyone. We're really honored to have you here. It's pretty amazing to look around this room. Of course, many dear friends who I haven't seen in a while because I've been goofing off at Carnegie instead of doing some real work in the lab. But it's been a lot of fun, and eventually I'll show you a little of the fun. I've made this transition about 1.5 years ago to joining this institution, and many of you know our department in Baltimore, our embryology department. But this institution actually is a lot broader and spans all the way from cosmology to ecology across this span. So as a sort of capstone experience in my life, I've taken up thinking about astronomy, cosmology, ecology, geochemistry, geophysics, and so on. And it's been great fun. For example, in the next slide, you'll see one of our giant telescopes in Chile. We have 2 of these, twin ones, and we have some other big telescopes that are building a giant one now. Fascinating work in the same way that it's the best time in history to be a biologist. Oh, you're getting ahead of me. Well, have a nice show. Well, that's fine. You could look at the lava of this volcano in Nicaragua, but I'll tell you about the telescope. You know, it's the best time in history to be an astronomer, just as it's the best time in history to be a biologist. And it's also an extremely interesting time to be a geologist, and we have a lot of them. And I accompanied these 2 volcanologists to Nicaragua to visit that volcano and see an extremely active lava lake. So pretty fascinating stuff. We have an airborne observatory, an airplane we own, which flies over the Amazon. And next slide will show you one of the images from Greg Asner and his team looking at the Tambopata River and one of its tributaries. This is high throughput ecology from the air, bringing to ecology more or less what high throughput genomic sequencing has brought to biology. So this plane just spent 2.5 months in Borneo. We're going to Ecuador next month. This is really fun. But of course, at heart, I'm a biologist, and so I'm trying to look at the unity of biology and genetics. And in the next slide, you'll show how I, conceptually bring this together when it's quite clear that a powerful genetic organism has deep-seated roots that have been revealed by the Hubble Space Telescope. So this is the unity of science that we're seeing. So you've just heard from 2 people who are now dear friends of mine, and it's important, I think, to realize the way we felt then, which was there was this scary other group on another continent doing things that might be the same things we were doing. You know, competition is a necessary evil in science, but it is often an evil because it separates people. But remember, what happens when you go to the conference and you have the right attitude is you meet your competitors, your direct competitors, and they care more about your work than anyone else on the planet, and vice versa. And you both have something to offer. And all of a sudden you realize you can spend all your time gossiping about your advisors and loosen up the tension tremendously. And sorry, Tom. And and have a great time together. And we did. We had a great time together because it was such an exciting time and led to lifelong friendships. So important to realize that. So I want to start in Indiana, postdocing with Tom Kaufman and Barry Polisky. And this, you know, we celebrate now all the time interdisciplinary science. Well, many of you do interdisciplinary science, and Barry was an expert on DNA replication in E. coli, who taught me how to clone DNA and manipulate it. And Tom, it was a pilgrimage to work with Tom. He was one of the great geneticists of the time. He discovered these amazing genes. He had insight after insight. There were many properties of the Antennapedia complex different from the bithorax complex. So both the parallels and the differences were fascinating. So fantastic person to learn from. The team that isolated the genes, and this was the required dress of the time, were Bob Lehman, who was a technician in the lab, and Amy Weiner, a graduate student, and that other guy who I don't recognize. And we spent 3 years isolating 300 kilobases of DNA, which is a little depressing to reflect on, but it was a really good 300 kilobases of DNA, and led us, and Tom's huge investment of work and his students, Barbara Wakamoto, Tula Hazelrig, and others, Ken Kempfuse, had really set the stage for bringing this other discipline of molecular biology to bear to try to figure out how these things might work. And that led, of course, to the Homeobox. So then I moved to a place with a really good view. Next slide, out my office window. This was Boulder, University of Colorado, and was joined there quickly by 2 postdocs who are shown in this next picture, and that's Alan Lawn and Sean Carroll, and some of these other folks came a bit later. Now, I recommend to anyone starting a lab to have 2 people of the caliber of Alan Lawn and Sean Carroll join you. That's a really good way to start and a good way to continue. I was extremely lucky, as I have been through my whole career, in having people working in my lab who I learned tremendously from, and I'm still learning from. So here we were with the Flatirons, and we were looking at genes and isolating genes. Sean ran a division of the lab called the Bay of Pigs. It was one of the bays. It was the messiest bay, vying with Levine. Next slide, I don't have a picture of that, but here's Sean celebrating his Boston roots, and Anne Boulay, who joined us a little bit later to work on some of the early genes, and then various others. Next slide will just show a few pictures of other people who came along fairly early in the period of the lab. I don't have a picture here of David Bilder, but he joined us a bit later at Stanford. And next one is John Tampkin, who with Jim Kennison brought us into working on chromatin regulators. You'll notice John's attitude about his future, which didn't turn out to be true. He ended up at UC Santa Cruz as chair of the department, but at the time, things weren't looking so great for all of us, but then the field exploded. So next slide shows one of the more important members of my lab, my dog Mariah, my first Samoyed of 5, and now the lab pretty large. And at the right, Joan Hooper. Can you go back, please? Joan Hooper, who eventually cloned Patched and got us going on hedgehog signaling, the whole next phase of the lab. So it was kids in a candy store, of course, thanks to many people, but particularly Nusslein, Volhart, and Wieschaus, who had provided the whole community with hundreds of different important mutants. And if you thoughtfully went after some of them with molecular methods, sometimes you learned something the geneticists didn't already know. Quite often, instead, you tested ideas the geneticists had come up with, and that sort of interdisciplinary work was tremendously productive and exciting. So Sean had been an immunologist in his graduate work. In the next slide, we've seen some nice in situs from those times, and here were some stains with antibodies showing the different distribution of homeotics and the famous Antennapedia mutant. And then in the next slide, we have also using antibodies, the assembly of essentially the Nusslein, Volhart, and Wieshaus hierarchy of segmentation genes as embodied in the patterns of expression of the various, or production of the various proteins. You can start to see that gradual refinement from very broad patterns to very precise and narrow patterns. And I'd like to be able to stand here and tell you with a straight face, we now understand how this works. And isn't it wonderful to look back 30 years and think how much we've learned? And actually, I think it would be more honest to say that many of the fundamental questions we had then, we still have now, which is embarrassing but true. And how the genome is translated into morphology remains a very challenging problem, as many of you know, because many of you work on it. It's also exciting to have such job security with a problem none of us can solve. So of course, in the isolation of these genes and the molecular work that was going on, things got kind of competitive. And I'll show you how extreme that got. I went outside my house in Boulder one time and saw this real estate sign, which you can see on the next slide. Now, some of you may not be able to read the fine print, but I was really startled when I saw what was going on. And all sorts of methods work in science. This may be not the most standard one, but here it was. This is not faked. This sign was really there in Boulder. So you've heard stories about the cross-hybridization, and I, of course, went through a similar experience working in Tom's lab and in Barry's lab, finding, of course, when you find a result like that, the first thing you think is contamination. Your probe is contaminated. And that's true with almost all exciting findings I've ever had in my career, or people in my lab have had. When you first see it, it's either completely bogus or extremely exciting, and you go through some sweaty times trying to figure out which of those is the case. Now, the cross-hybridization stood up, and then there was the issue of having to sequence. And at the time, there wasn't too much sequencing going on in our department at Indiana, where I was then, but Tom Blumenthal's lab was doing some. And they showed me, and I isolated little fragments that we now know contain homeoboxes and ran my first sequencing gel, and it looked awful. Of course, it was P32. It was a 14 by 17-inch piece of film that I developed at midnight one night. And there were all these lanes that wobbled back and forth across the film and had blank places and smeared places and everything else. But there were some sections of readable sequence. So what was it? At this point, we didn't know whether these genes were protein coding. We didn't know whether these genes were genes. We knew so little about all these things. And this was FUTS and Antennapedia and UBX, so in my case. So the question was, what was on this gel? And it was midnight, and nobody was around, and I couldn't find the genetic code. This is very frustrating. You're sitting there, and you're likely to have a really cool result, and you don't remember the genetic code. You can remember U, right? That's the first one, right? Beyond that, you're sort of, oh, geez. So then you start running all over the building. Where can I find the genetic code, right? You don't look it up on the World Wide Web. And I went to the library of the department, and of course, there are all the journals, but journals don't have the genetic code in the, in the front of the journal. It's not that standard. And eventually, I found the 1966 edition of the Cold Spring Harbor Symposium on Quantitative Biology, the big book. And that was the genetic code year, and they hadn't figured it all out, so there was still uncertainty about one of the stop codons. But I didn't want stop codons, I wanted go codons. So I sat there and started translating in all three frames, each of these sequences, and then suddenly there was a little similarity, just a few amino acids, and suddenly that was a great moment and that was about 1 in the morning, and there was nobody to tell except a rat. There was a rat, but I didn't explain it to the rat. So suddenly you realize there are protein coding genes, there's protein coding similarity between a segmentation gene and a homeotic gene. There's similarity between the bithorax and actinopia complexes. Ed Lewis and Tom Kaufman were right. Everything comes suddenly in that moment. So we're lucky, any of us, to have moments like that. I've had one, but I'll take it. I'll take it. It was great. It was really fun. So that led, if we go to the next slide, to a couple of papers. This one on the homology between these genes and the suggestion that maybe they are related to each other functionally in some manner. Communicated by Bill Wood, who I saw this afternoon. It was great to see Bill again. He was our initial chair at the University of Colorado at Boulder. So that was exciting. And then in my lab at Colorado, Alan Lawn did a very careful study, the first segmentation gene sequence of the Fushitazu, the whole thing. That led to the paper on the next one here, which was about the FUT sequence and so on, and temperature-sensitive alleles. And Alan noticed that this homeodomain sequence, and this was independently discovered in Walter's lab, that this homeodomain sequence looked like yeast mating type protein sequences. And I remember our reaction was mixed. On the one hand, wow, yeast mating type is the best studied molecular biology system of a eukaryote by far, the most interesting thing. Ira Herskowitz and many others. And the second thing is, oh my God, we've got now we have to compete with all those yeast people. And they're still here. They're just down the hall here. They're still after us. It's terrible. Anyway, but of course, it's a fabulous moment to suddenly see the biology connect like that. And Sandy Johnson, who bought the homology right away, was fascinated by the thought that this was a billion-year-old DNA protein sequence and structure that docked against DNA. So this was very exciting. And again, the first blush, you think, okay, we've got it now. These things control batteries of subservient genes, and those genes then give the different segments their different features or whatever. And if only we really understood that, But it is a beginning. It's a very important step, I think. It's very exciting. But we still don't understand how coordination of target genes gives you morphology. And we'd love to, so we can understand, for example, our vertebrae and our fingers and so on. So then the work that Bill just mentioned, showing that these ideas applied across the whole or most of the animal kingdom were incredibly exciting. The next slide, just a celebratory picture, this is my last slide, of the sort of unity of developmental biology that we all enjoy now. Suddenly there was a common language between people who worked on very different organisms. I don't know how we were ever befuddled into thinking that different animals would use very different control systems for development. There really isn't anything written down a long time ago that said there must be common genetic hardware for controlling development. Usually it's like the Bible, you can find a metaphor for anything, or some loony who said it at some point, right? But if you go back in this particular field, people were not saying that the genetic hardware would be conserved so much among animals, and yet it's impossible to imagine anything else, because when is a referee going to blow the whistle and say, okay, new system, let's start over. So this emergence with Tin Man and Eyeless and so on, all these things happening, gave a sense of momentum, a sense of excitement, a freshness to the field that has kept going right through today. And this group here is a celebration of that too. There's so much more to be learned and figured out and enjoyed as this goes forward. So we were lucky. We had this, the three of us had this wonderful time with great mentors, a tremendous fortune in being there at the right time, the tools at hand that we needed, tools invented especially by David Hogness and also by others. And these were things that all came together in a very interdisciplinary way to lead to discovery and another step toward the kind of understanding that we're all still working toward having. So thank you for your attention.
Thanks so much to all three of you, and thanks, audience, for your enthusiasm and for laughing at all those jokes on cue. That was great. Sorry, it was very serious. We don't have a ton of time. I don't want to keep you guys for a long time. But what I would like to ask is a couple of questions of you guys, and then hopefully we have time. I know that you have some special stories that you want to tell that we can consider them bedtime stories or pre-bar stories or whatever's appropriate for you. So the first question that I am wondering about, we've talked about how it's a very exciting time right now. There's no better time to be a biologist. At the time, in the late 70s and early 80s, there was so much phenomenology about these genes and the phenotypes that they caused, and you were able to put sort of a molecular face to it. 30 years later, we understand the commonality of the genome and conserved processes for controlling development. What else do you think we need to know about the homeodomain at this point?
About the homeodomain per se?
Yes, the homeodomain per se, if anything.
I don't know anything about the homeodomain per se. I think there's still a lot of specificity in it that's not completely understood, and a lot of protein-protein interactions that aren't known on the back of the homeodomain, and how those affect regulatory specificity. But I think the major questions are still how you control or how you vary morphology with different homeotic proteins. There's a lot that's been learned, but there's a lot more to learn. So.
Yeah, I mean, the Hox proteins are really very wimpy. I mean, they typically bind as monomers, micromolar concentrations. It is amazing, the conservation is amazing, the conserved role and patterning, the head-tail axis in so many different animals is kind of surprising to me because on their own, there's just not much there. So there is a deep combinatorial code that has still managed to evade us, I would say, up to this point.
Okay. My next question is not specific to the homeodomain, and I hope that's okay. It's interesting to me that all three of you who were involved in this very important discovery have now taken up very significant senior leadership positions at the heads of major national institutions. I'd like to ask if this was something that you planned in your career, and whether that is the case or not, at what point in your career did you transition, if such a transition occurred, from thinking only about your own experiments or the experiments of your trainees to essentially what you are directing now, which is a future vision for biological research in this country?
Okay, well, I'll take a turn. At Stanford, I became head of a program called BioX, which is biology-centric, but involved 50 departments at Stanford, 400 faculty. We put a building together with 44 faculty from 25 departments. And that was just incredible fun. And it meant that biology could go in all sorts of directions quite rapidly in ways that it would not have gone with the typical segregation of departments into separate buildings or even separate areas of buildings. And that was just a fantastic experience, seeing the synergies just erupt right and left, with the most important roles being played by the students who teamed up and postdocs who teamed up with people in other labs and taught their advisors how to talk to each other, which was absolutely crucial. That also created whole new niches which could be occupied then by those students and postdocs, so that was great. So giddy with that, I said yes to this with telescopes and lava lakes, and that's a bigger stretch because it's no longer biology-centric, but we're all geneticists probably in this room, and what we do in genetics is we turn over rocks, basically. We look somewhere, we don't know what we're looking for, we just want to find something that matters, and that's what astronomers do for sure, and what I'm going to northern Canada at the end of the month to look for 3 or 4 billion-year-old rocks with some of our geologists, and that's literally turning over rocks. So that discovery process that starts an intellectual inquiry is common to all these fields and tremendous fun to do. Now, I have to say, I don't think I'd enjoy my present job if I hadn't spent 30 years doing more serious science, working closely with a wonderful team of students and postdocs, rather than doing my sort of dilettante thing now. But as a capstone experience, it's really been quite wonderful.
I never dreamed of being an administrator. I wanted to do science. And I still love the process of doing science, getting up in the morning, coming to the lab, yelling at everyone, thinking up new experiments. I mean, it's just the process of doing science is a wonderful thing. And right now, as far as me, I mean, I've been at Princeton, as this director thing for less than a year, and I'm still I have not completely transferred my ego from the lab to the greater institution. Now, I know they're going to start, don't tell anybody this, because, I am supposed to deliver something to the institution. I understand that's why they hired me. But don't give up being a scientist. It's the greatest thing.
I would say I don't know why I'm a dean right now. But it does allow me to segue into a story about Mike Levine, who always had a terrible problem with authority figures. Terrible problem with authority figures. And Basel was an incredibly exciting time, many people visiting, and there was a time when Dave Hogness visited. And we were in awe of Dave Hogness. This guy dominated developmental molecular biology. He was a giant. And we were invited to a dinner with Dave at Bernard Meckler's house. Mike and I were stuck, at the very end of this table, almost out, overlooking the Rhine River, almost on the balcony. And the conversation was going on. As usual, Mike was trying to get a word in edgewise into this conversation. So Dave Hogness and Bernie Meckler were dueling back and forth and trying to tell one story that was better than the other. Mike kept trying to get, a little anything in there, more and more frustrated. And he couldn't get anything in. And so finally, Bernard told some really interesting story, and Mike, at the end of the table, said, "Well, Dave, let's see you top that one." And the table went silent, completely. And Dave, who was a formal person, kind of swiveled his head toward Mike, and Mike, kind of tried to joke, "Hi, Dave, we're real buddies." And it was clear that Dave didn't think they were real buddies. And the upshot of it, poor Ernst Hoffen, our buddy Ernst, was sitting next to Dave. And his whole reason for being at that dinner, he was going to ask Dave if he could do a postdoc in the Hogness lab. And his face fell completely, because he realized, being at that table after Mike had done this terrible thing, his chances were over. So now Mike is the authority figure.
Whoa. So that was your segue, from, he doesn't know why he's dean, and the next thing you know, you get a Levine story. Okay, I got a McGinnis story, and there's no connection to it, but here we go. So he showed that zoo blot, all right, which many of us regard as kind of the beginning of the modern era of Evo Devo. All right, there it is. Now, he and his assistant Nadine went out and collected all sorts of slithering creatures. They didn't really know what they were. And they would always get two. It was kind of a Noah's Ark thing. They get two. One was ground up for the blot that you saw, and the other was put in a baby food jar with a lifesaver. For some reason, Nadine thought that was a very favorable carbon source to grow all these slithering creatures. And Bill, labeled the lane, and he even showed this lane. It was originally labeled worm. And the worm, I looked at the other copy in the baby food jar, it had arms hanging out of it, which is very, this is the father of Ivo von Devo here, thought it was a worm. And I said, look, I'm from Tarzana, California. I've never really seen a worm, with arms hanging. I mean, these were like gnarly looking appendages hanging off this worm, right? And one morning, Nadine thought that the duplicate copy of the worm that had been sacrificed, for the blot, was dead, but it eclosed one morning. She shrieked. It was a beetle. And so I said, Bill, your worm became a beetle. And being unflappable, the great Evo Devo biologist simply erased worm and replaced it with the word beetle. And that's what you saw.
It's so lucky I don't have any of my immediate colleagues here to tell these stories about me. It's good. I'll mention something about the importance of friendship, though, since we've been talking about it. Even a friend you didn't even know was a friend. So I was sitting there in Indiana trying to figure out how to do any of this kind of chromosome walking and so on, far from David Hogness. And Welcome Bender was headed to Harvard, driving from California to Harvard to set up his own lab. And that's a time of high pressure, and you're worried about everything, grants and setup and getting students and all this. And Welcome knew I was starting up, and he took the trouble with extreme generosity to pull into Bloomington, Indiana, and spend a couple of days visiting. Of course, part of it was to talk with Tom about things I didn't yet understand at all, so it was great for me to listen to them. But he also very generously spent endless hours with me going through the details of how he did his procedures for chromosome walking, which got me off on a tremendously strong start, right? And he could have viewed me as some sort of competitor or something. Instead, again, lifelong friends. So fantastic importance of friendship and generosity in science. That's yet another of many examples. I know all of you have your own examples.
That's great. Well, I think that's a fantastic note to end on. I think, we've laughed and we've cried and we've learned about the Homeobox in more detail than we knew was possible. I want to really thank Bill McGinnis, Mike Levine, and Matt Scott for not only their great contributions that literally enable the work that many of us do here, but also for their generosity in sharing not only their scientific process, but some of their personal process with us as well. So thanks very much to the three of you.